Thank you, Madam MC. I would like to thank the organizers, all the organizers, but especially Dr. Balarao and Dr. Yakmi, who work behind the scenes on many accounts to make it possible for me to come here. What I would like to share with you today is the way that we do electron microscopy on liquid systems. And this is something that is really against the nature of the microscope, because the electron microscope works under high vacuum. So what I will show you today, first of all, is how to make the specimens compatible with the electron microscope, both the scanning and the transmission electron microscope, and how we also are able to make sure that what we see in the specimen really reflects what you have in the vial from which you have drawn your material. Before that, on this first slide, you see a view, a panorama of Haifa. Some of you have seen it. This is looking north. We are built on the slope of a mountain, Mount Carmel, and we are located on the Mediterranean Sea, about 100 kilometers north of Tel Aviv. Tel Aviv is better known. I'm a member of the faculty of the Technion. The Technion was the first university in Israel, and this year we have celebrated the 100th anniversary of laying down the cornerstone. Now, before I forget to do it later, I would like to thank all the good people who gave me money to support the things that we do so happily. And even more important, I would like to acknowledge the technical help that I get from three wonderful ladies, Judith, Ella, and Berta, who have been with me a very long time and who really make many things possible, supporting the students and myself. So basically what we are after is, what I'm trying to describe today is cryo-electron microscopy. And basically this is just electron microscopy done at low temperatures, and I'll show you why. The other thing that I would like to emphasize is that you see here in parentheses the first line, you will see we usually call it cryo-EM, but nowadays most people are starting to call it cryo-EN, because we are after nanoscopic details, and because of that, the proper term would be electron nanoscopy and not electron microscopy, a term that has been carried on since the 18th century. So I'm going to talk today about cryo-TEM and cryo-SEM. Freeze fracture replication is something that I'm not going to describe today. The objectives that we have is basically to obtain direct images. That is, we want to look inside the material and understand what's going on the supramolecular scale. That is, we would like to get direct images. We actually see them with our eyes. It's not just collecting a spectrum, for instance. And we would like to have resolution that is on the level of how molecules arrange themselves into aggregates. And I'm, of course, going to give you several examples. Quite often, we also want to see how system parameters affect the structure that we see in those liquid systems. Now, structured liquid systems are all around us as inside. Just think about the body fluids. All of them are nanostructured liquids. And if you think about shampoo and liquid soap, that's also a nanostructured liquid. So these are not rare materials. They are really everywhere. Below here, you can see, on the lower part of the slide, you can see some of the structures that we have looked at. Okay, so basically what we want to do is we want to make the specimen, which is liquid with a lot of movement and high vapor pressure, we want to make it compatible with the electron microscope. And to do this, we have chosen the route of very quickly cooling the liquid that we have. Many biologists still use another class of technique by which they add a stain and then they dry. But this is very difficult to preserve the specimen nanostructure with. So we are taking not the chemical route, but the physical route, cooling very fast and then looking at the cooled specimen. Now, when you cool a liquid, you get an ice. It can be a water ice or something else. But here, much of the work that we have done has been done with water ice. And the challenge here is that when you cool water, typically you get the kind of ice which is, oops, I'll have to get used to this, the kind of ice which is marked here by IH. This is hexagonal ice. This is crystalline ice. IC denotes the cubic ice, which is another crystalline ice. But what we would like to do is cool so fast that the specimen does not crystallize. We want to make a supercooled water, a glass. And this is, you see it on the screen, in areas that are denoted IV, vitreous ice. So we want to make it a glass. This is not a real solid. This is a liquid of very low vapor pressure and very high viscosity, and it preserves the original nanostructure of the system. The problem is that in order to achieve this for water, and many of our systems are aqueous systems, you have to cool very fast to the tune of 100,000 degrees per second, 10 to the 5th degrees per second. But this is feasible, and the way we do it is I will explain. I will not bother you with the details, the technical details. Those you can see in our many publications on the area. Another challenge of these systems is that they are very sensitive to temperature and concentration changes. So you have to make sure that during specimen preparation, you make them in a box like this one here, which we could call the controlled environment vitrification system, in which you control both the temperature and the saturation of the gas phase. If you do that, you ensure that the structure that you capture is that you have in the vial that is of the one of interest. And I will show you later on towards the end how important that is. The box that you see here has a very significant Indian connection, because this work was done with a very bright graduate student, Jayesh Bellary, now Professor Jayesh Bellary of IIT Bombay, who is here with us. And back in the 80s, we developed that system. And as you will see later on, we took that system and even took it to different directions. So this is, but on the screen, you can see the first publication going back to 1988. So going very close to 25 years and still going strong. Okay. So basically, I will just describe it in words, and I will not go into details because we have no time for that. You take a liquid drop, you put it on a support film which has holes in it, and you make very thin liquid films out of this liquid drop. The film is about 20 to 300 nanometers thick. Because it has such a very high surface area to volume ratio, when you quench it in a cryogen, the right cryogen, for instance, for water, we use liquid ethane at its freezing point. The cooling rates, and we have measured it, are indeed 10 to the 5th degrees per second. And the samples, as you can see on the screens here, the samples are really vitrified. So quickly, what do we see here? First of all, this is an example of a self-assembled system which is quite complex. It is made of monooleuin, which is sort of a lipid, and a non-ionic polymeric surfactant, pluronic F127. And it forms here in the conditions that you see here, and this is work that we did with Maura Monduzzi and her group in Cagliari in Italy. You see here on the screen, you see 3 different types of structures. You have this one here where the arrow is, the cursor is, this is a lamellar phase. You see this thing here, which is a cubic phase, a liquid crystalline cubic phase. And then you have the intermediate, which is this thing here with the white arrows. So we have, in one field of view, we have 3 different phases, all clearly seen by the electron microscope. This is really one of the main advantages of electron microscopy, that you don't need a model to interpret your data. And especially when you have a mixed system or a system with a very wide, very broad size distribution, it would be very difficult to interpret the data if you didn't have a model like this. 2 more comments. The dark thing that goes here in the bottom is part of that support film that I told you. And the other very important thing here is where the cursor is this is the vitreous ice. This is super cooled water. So you see the water actually did not freeze. It just cooled very, very fast, preserving the original nanostructure. People always ask me, what are these black dots? These are the most having the best contrast. This is just little specks of ice that land on the specimen during preparation. We try to avoid it, but you cannot do it always. Another comment I would like to make is that it's not that we don't use other techniques, but we always try to use both the microscopy, the direct imaging, and indirect techniques, like scattering, X-ray scattering, neutron scattering, electric conductivity, and so on. The reason for this is that microscopy gives you the building blocks of your system. With those, you can build a physical model with which you build a mathematical model to analyze your data. So we always work together with the two techniques. A quick biological example is given here. This is work that we did with Sharon Golan, a former graduate student now at MIT. And what you see here are complexes of DNA and lipids which are used. These very nice ordered onion-like structures are used to infect cells with new genes. So this is used for transfection, in other words, for gene therapy. If you don't form these structures, it turns out, the DNA or the RNA will not go to its target nucleus. So this is a way to do it, but of course I have no time to elaborate on that. She has seen it also in a completely synthetic system like this one. It turns out that these onion-like structures are very predominant in interactions between negatively charged and positively charged surfactants or polyelectrolytes. While water is very important, probably the most important liquid not in the universe, but at least on our planet, there are other liquids that are of interest, and here I'm going to share with you one which is really showing the extreme that you can go with cryo-electron microscopy. What you see here are nanotubes, carbon nanotubes this time, suspended in a superacid, in chlorosulfonic acid. And what you see here, these are fairly large. I chose fairly large nanotubes so everybody can see what I'm talking about. These are these things here. But you notice that some of them are white and some of them are dark. Some of them are dark, some of them are white. Dark, white. So have a look. What do we do this for? And this is connected to some way to what was already described earlier. What we want to do is take these carbon nanotubes and spin them by themselves without any additions into fibers. And in order to do it, you have to first suspend them in a solvent. And it turns out that the only good solvent is this chlorosulfonic acid, which is a really nasty material because it will oxidize, it will disintegrate. When it disintegrates, it will release hydrochloric acid and sulfuric acid, something that you don't want to play with at home, believe me. But without going into details, we have developed a methodology that allows us to prepare safely good specimens like the ones that you can see here. This is work that we've done with Matteo Pasquali at Rice University, and we have published strings of papers. The last one is to be published soon in Science. And we are able to prepare specimens without harming the specimen, without harming the microscope, and without harming the person who handles them. Not necessarily in that order. Okay, so we are doing it quite safe. And the reason now we do it is I'll show it in the last part of my talk. We eventually want to see how these nanotubes arrange themselves into pneumatic liquid crystals from which we spin the fibers. Okay, so this is TEM, this is cryo-TEM, and we have done cryo-TEM, and Jayesh can attest to that, for over 30 years. But more recently, we have been able to extend our studies to cryoscanning electron microscope. In scanning electron microscopy, actually you look at the surface of a fracture and you don't look through a thin specimen. That's a very different instrument. Actually, while the TEM is an optical instrument, the scanning electron microscope is a scanning probe microscope. It's not an optical instrument. But the question is, if TEM is so great, why bother with another technique? First of all, quite often you want to use both techniques to rule out artifacts. That's very important. And also, quite often you have systems that are too viscous to make into the thin films that I described before. In other cases, you have large objects, larger than 0.5 micrometer, for instance, in which you want to identify nanoscopic objects. For this, you need the scanning electron microscope. And what you see here is the relatively new state-of-the-art Zeiss Ultra Plus high-resolution microscope. It has a field emission gun, and that is needed because in order to do high-resolution SEM, you have to use low voltage. And if you want to have enough signal at low voltage, you need a field emission gun. It also has very good detectors and very good optics. This is just an example. This is one of the first examples that we got with this instrument. You have here a titanium mesophase. Do pay attention that this was taken at original magnification of 200,000 times, very high for SEM, and at 1 kilovolt, of course. But room temperature, high resolution SEM is a child's play. How do you prepare specimens for cryospecimens, liquid specimens for high resolution SEM? So basically what we do is we take a liquid, we cool it as fast as we can, and we fracture it. Here's one example. What you see here is a sandwich made of these two gold planchettes. This is a schematic. You have a liquid, a few micrometers of liquid between the two. You plunge it into the cryogen, and you open it. You open the sandwich. By opening the sandwich, you get two fracture surfaces. And then, if you want, you can sublime some of the volatiles in order to get better topographical contrast. And if you want, you can also coat with a thin metal coat in order to make the specimen conductive. This is actually not needed if you use the right low voltage, because if you use the right low voltage, the number of electrons hitting will be equal to the number of electrons emitted, and then you don't need conduction in order not to charge your specimen. So you have to really select your parameters very carefully. I'm not going to describe all the gory details that you see here, but I just want to say, and Jayesh, this is for you. This is a paper. The other one was in 1988. This is in 2012, this year, and we published it with another brilliant graduate student, Liron Isman. And this is the box in which we can prepare cryo-SEM specimens under controlled conditions. I'm not going to go into details, of course. This is the little specimen holder that allows you to actually take the sandwich and fracture it. The fracturing itself is done in a special apparatus where at low vacuum, at very high vacuum and very low temperature. The specimen then is transferred with the shuttle. This is the shuttle to the electron microscope. Now you can see the shuttle attached to the electron microscope. The shuttle is evacuated and kept at low temperature, so the specimen can be introduced, and now you look into the microscope, it can be introduced without any harm into the electron microscope. And this is what you see. So this is an ointment, a medical ointment, and what you see here is a structure that is called a cochleate. You see, this is a cochleate. What you see here is a bilayer of lipid which is wrapped around itself. And if it will let me, if we go along this cochleate, you see here a step, and then another step, and then another step. Each step is a bilayer of lipid, a swollen bilayer of lipid, which is about 7 nanometers in thickness. Another thing that I want to draw your attention to, that we use here two signals. There are several signals that are emitted from the specimen when the beam hits it, and we have here, and it's basically backscattered, X-ray, and secondaries. Here we use two different secondary electron detectors to give us the very nice high resolution with the three-dimensional appearance of the objects that you see here. Very briefly, another challenge are microemulsions. Microemulsions are mixtures of water and oil. Of course, they say that water and oil don't mix. Well, they don't mix if you don't put inside some surfactant. And if you put the right surfactant at the right temperature, they will mix and form a microemulsions where you have small domains of water in oil or water in oil. And in some cases, you have actually a bicontinuous phase. Here is an example. This is based on water, octane, and the non-ionic surfactant C12E5. We're going to see two examples. One at here at 80% oil in the oil-water mixture, and one in the 32% oil to water. And of course, there is difference in temperature. And I have to show you, I would like to show you, if I can get the cursor, that all this channel here on the phase diagram, this is a very peculiar phase diagram, but still a phase diagram. All this is a one-phase microemulsions where one structure changes into another. And here it is. Alpha 80, that is, we have 80% oil in the oil-water mixture. And what you can see very well here are very small domains. Those little blobs are small domains of water in the oil. If you are not careful and your temperature goes 1 degree off, you get a very different structure. It phase separates and you get a lamellar structure. So you see this is 38, this is 37. So very good control of your temperature, which is afforded by the controlled environment vitrification system, is really essential. If we go now to the alpha 30 and temperature of 31, you see that we get the sponge-like structure, which is characteristic of a bicontinuous phase. And this was something that we tried to do for a very long time. And in fact, now we can do it both by TEM as well. Here you have the same structures by TEM. This is the alpha 80, and what you see here are small droplets of water in a continuous phase of oil. The water droplets are sometimes dark, sometimes white. The reason for this is that this particular specimen was cooled in liquid nitrogen, which is a very bad cryogen, but it cannot use for this one liquid ethane, which will dissolve the octane. However, the octane that we used here was branched octane, and it vitrified very nicely. You see the background is featureless. However, the little water droplets freeze. They froze, and some of them are aligned with respect to the beam in such a way that they scatter electrons while others don't, and that's the difference in the appearance. And this is the bicontinuous one. And in fact, that we can do this by two different methods and get the same structure really indicates that we have been successful to capture a real object and not an artifact. So whenever we can, we use more than one technique. Another microemulsion, and I want to let you know that I've been talking now for 23 minutes, and I intend to be on time, so no reason to despair. Another example is here. It's an old problem. It's a problem that has been in the literature for 25 years. It was a system that was studied by two giants of colloid science, Barry Ninham and Fennel Evans, and they found out that if they take a double-chain surfactant, didodecyl dimethyl ammonium bromide, and mix it with water and with oils, different oils, at the right proportions, they get large domains of microemulsions. But they did something else. What they did, this is stuck now. Oh, it's here. Okay, what they did was they measured the electrical conductivity, or rather the resistance, as function of the addition of water. So if we look at this one here, octane, which you see here at low water concentration, the conductivity is high, the resistivity is low, but as you add water, the resistivity goes up enormously, orders of magnitude. Now, if you think of it, this is really unusual. You add water, which is conductive, and the system becomes no conductive. This is counterintuitive, right? And they came up, since they're very good people, they came up with a model which shows that at low water content, water forms a channel, a network of channels, of tubes, which span the entire system. And this is what gives it its conductivity. It's surrounded by oil. However, when you add some water, this structure breaks up, and what you get are individual, separate globules of water surrounded by oil. The water is not continuous. The water is only the oil is continuous, and that's why you get a high resistivity. 25 minutes. You hear that? Okay. So have another 5 minutes, which I'll use about 3 of them. Okay. So can you see it by electron microscopy? Not until now, but very recently, and we published it just a month ago. We can see that exactly the same structure. Look, this is the electron micrograph. This is the model. It shows 2 things. First of all, that those guys were really good. And the other thing, that the graduate student who did it was also very good. His name is Ido Ben Barak. But this is not enough. Now let's add some more water and see what happens. And this is what happens. As you can see here, the structure is completely different. You have oil, you have water droplets surrounded, I'm enthusiastic about it, water droplets surrounded by oil, and this is exactly it. Moreover, if you take an intermediate concentration of water, you get the intermediate structure, okay? On the lower left side, you see the transformation from water to oil. I'll take 1 more minute to show only, no, I have to take 1.5 minutes, I'm sorry. I want to show you that we can do also carbon nanotubes. And before that, I showed you a low concentration of carbon nanotubes. In this picture here, in the lower part, I show you that when we go to 0.25%, we get a very viscous system, which we can still make into a TEM picture. You see, this is still a TEM picture, the lower part of the picture. But it's getting really very viscous. At 0.5, you cannot make it, it's becoming liquid crystalline, but you can see very well here now the order of the liquid crystals. So this is also extreme cryo-SEM. This is in chlorosulfonic acid, and this is due to the great assistance, the great work of Olga Kleinerman, who is an MSc student going on PhD. Last example. That's it, I promise. What you have here is I want to show you that you can use you may use not only secondary electrons, but also backscattered electrons. And this is a nice example. You have here a dispersion of 2, I hope you see it, two ceramics, alumina and yttria in ethanol. And what you can see here is the vitrified specimen. And this is a backscattered image. And you cannot tell here from this what is aluminum oxide, and what is yttrium oxide. But if you switch to the backscattered electron, and there is a very good high-resolution backscattered electron in this microscope, you can see very well two types of particles, bright ones and dark ones. The bright ones are that of yttrium or yttrium oxide, because yttrium is heavier than aluminum. It will give off many more backscattered electrons. So don't forget that in most microscopes you have or you can have backscattered imaging, and it's really useful. As promised, I'll finish here. I hope I was able to show you some of the good things that you can do with cryo-EM. I also gave you some warnings. One warning that I haven't given you is that all the systems that we have shown here are very sensitive to the electron beam, so they can be easily taken apart by the beam, by radiolysis. So all the images that you saw were taken at conditions of very low electron dose. And this is a precaution that you have to do whenever you look at these systems. So again, thank you for the invitation. Thank you for the opportunity. Thank you for listening so nicely. Thank you. I guess we have a few minutes for questions, I hope.
You know, Ishi, very nice talk. I wanted to ask you a question on the last few projections. You know, this Ivan and the other good gentleman you said, they had the model, you have the water channels, and if you raise the concentration of water, they become globules of water and less conductivity. Simple. Now, if you raise the temperature of the same system, what happens? Without raising the concentration of water.
Well, quite frankly, I don't know. They didn't do it. We haven't done it yet. But this is very, very fresh, and we actually, it's something that crossed our minds as well. But the honest answer is that we don't know yet. But you know, the whole thing, this is something that it's a general, the phenomenon is general, and what you have here is a consideration of packing, basically, of the molecules. So curvature and energy of packing.
I have one question here.
Thank you very much, first of all, for your talk. Very nice and interesting. I wanted to ask you, what do you mean when you say to prepare fibers from carbon nanotubes without polymer?
Yeah, I knew that it will tickle you a little bit. Okay, this is, you see, the high strength polymers like Kevlar, for instance, which is used for bulletproof vests, are made in the very same way. You disperse them in a solvent, and then you anneal them in such a way that you take the solvent away, and by van der Waals forces, they stick together and become very, very strong. So in Kevlar, it is done with sulfuric acid or phosphoric acid. In carbon nanotubes, we have to do it with chlorosulfonic acid. And I wonder if we can't do it also with the, no? Okay. All right, I thought about it, but maybe not. You can do it, by the way, with boronitride fibers, and we have a paper in preparation now about that. You can dissolve it in boronitride nanotubes. You can dissolve in chlorosulfonic